Hello students, how are you? Today we are going to discuss the next part that is the competent host. That is the third tool of the genetic engineering. So the next tool here is the competent host. Okay, competent host means the organism, the host body within which our rDNA or the vector which is with the foreign gene will able to multiply or able to produce the product, that is suppose protein. So just see, these are the host. What are actually the host? The host which can carry a foreign gene. Okay, and easily the foreign gene can be inserted inside the body, so that will be the competent host. Normally, generally, what type of competent host are used, that will completely be dependent on the type of experiment. So depending upon the type of experiment, a different type of competent host are actually selected. So first of all, see, the variety of hosts are used like bacteria, then yeast, plant cell or animal cell. Okay, so normally bacteria is used. Bacteria is very much important. It can be used as a good competent host. Why? The reason is that this bacteria will have very high rate of reproduction. It will easily multiply, so that's why bacteria are used. If it is not, then some fungi, yeast is taken. If we need to get, suppose the eukaryotic product, okay, eukaryotic protein you want, then inside the bacteria it will be not possible, because bacteria will provide the enzyme responsible for the prokaryotic, suppose, then prokaryotic enzyme will be released, which is not possible for the foreign gene to multiply, to incorporate inside the host body. If the gene, our gene of interest is eukaryotic and we want a host that is prokaryotic, then it is not going to match. So specifically, if we want a eukaryotic protein, then obviously we have to use the plant cell or animal cell. So it will be purely depending on the type of experiment, what type of product we want, or what is the main experiment, main aim of the experiment. Depending upon that, a different type of host are taken. So it can be bacteria also, fungi also, plant cell or animal cell. So what happen if it is a plant cell or animal cell, then there is a difference that will be always there, that if it is the plant cell, two membranes will be there, that will be cell wall and plasma membrane, and if it is the animal cell, then only the plasma membrane will be there. Okay. So just see, eukaryotic hosts are preferred if the eukaryotic proteins are required for if the protein we want to get, okay, then that protein is eukaryotic protein, then obviously that host will be also a prokaryote if the eukaryotic hosts are preferred for the eukaryotic protein to be synthesized. So if we need the eukaryotic protein, then obviously the host should also be a eukaryotic organism, right? Next one, as the DNA is hydrophilic, it cannot cross the plasma membrane. Okay, the main problem lies here, that suppose we have cut a gene fragment, right? We have cut the gene fragment. Then after that we have introduced into the vector, we have selected the vector, then after that we have inserted the foreign gene, we made one rDNA. Now this rDNA will be inserted inside the host body. So how we will able to enter or inject that rDNA into the host body? We have different methods. So that's why the different methods are used. Different methods will be based completely upon the competent host type. What is the type of the host? Depending upon that, we were going to have different methods. How we will inject the rDNA into the host body, right? So just see what happened. Different methods are here. So first method is some ions. These ions are the divalent ions. Okay. So what are the divalent ions? Like calcium. Okay. So if it is a calcium ion, suppose this host and rDNA, both of them, this is inserted into a medium having a divalent ion like calcium ion, then what happens? For after some period of time, the plasma membrane of the host will start to develop very minute pores, okay, and from the pore that rDNA will be inserted, okay. So this is one method. The second method is temperature fluctuation. So if we can change the temperature in that medium where this host and the rDNA is present, then also it is possible that the host can accept the rDNA, or rDNA can be injected inside the host body. Now the thing is that first of all, the temperature, suppose the normal temperature is 37 degrees Celsius. Now first of all, if we increase the temperature so that the host will not die, but at least it can little expand, okay. So the plasma membrane will expand if we increase the temperature. So first of all, we will increase the temperature so that some small pores can be developed, and whenever suppose rDNA is injected after suppose some interval of time, then what will happen, we will suddenly put the ice cubes into the medium so that suddenly the temperature can cool down. So suddenly the temperature will goes down, decrease, and the cell will start to shrink. And whenever the cell will shrink, then what will happen? Already our gene of interest will be injected inside the host body, isn't that? So these are some methods by which actually we can able to inject the rDNA, which is our main purpose, that we have to inject the rDNA into the host body. So how the host body will accept our gene fragment, that we are going to discuss in the method part, okay. So already you understand what is competent host. The host within which our gene of interest will multiply, or our gene of interest can produce the proteins, okay. So that's why different methods are here available so that we can inject the rDNA into the host body. So this thing is here, see different methods are here. From those methods, see chemical method is here, then gene gun or ballistic method, then electroporation and microinjection that are present. That should be the methods for the host to make a competent, or to make the host transform it, okay. The host should accept, the host should carry now our gene of interest. See, first of all, we have the chemical method, okay. So in this chemical method, what happened, first of all, this host, suppose this is the host, this host is taken into the liquid, a medium, and that medium is going to have one chemical. What is that chemical here? This chemical is PEG. PEG stands for polyethylene glycol, right? So whenever polyethylene glycol is in a medium, then the host is injected, then the host will develop some small pores, and in the same medium we have our gene of interest, that means rDNA is there. So whenever the cell will have the expansion or they develop the pores, our gene of interest will be inserted, okay. Normally will be also inserted, and the host will be a recombinant host, or that host will be a transformant host. So this is one method. Just remember that in this chemical method, one chemical is used, that is PEG, polyethylene glycol. So whenever polyethylene glycol is used, our gene of interest will be inserted, as the cell membrane will develop some minute pores. So that is the first one, first method. Second method is the gene gun method or ballistic method. This is very much important. Remember that gene gun method or ballistic method is very much important, right? See, so what happened, the DNA isolated with the DNA coated with the microscopic plate of the gold or tungsten of the size 1 to 2 micron are shot at the velocity of 14,000 feet per second. Now just see, we are going to have the host here, okay. So suppose we have the host here, right? Now with a tungsten, suppose here this is the force we are going to apply with a velocity of 1400 feet per second. Now we will take some type of metals which is non-reactive, like gold or tungsten, okay. Suppose these are the gold particle, okay, gold dust or the tungsten, okay. Now this gold dust or the tungsten is going to have our gene of interest, okay. That means this gene of interest will be coated, okay, right. So what will happen, our gene of interest or that whole vector recombinant DNA is coated with the that gold or tungsten. Why we are using the gold or tungsten? Because it is going to be non-reactive as it is a metal. Okay, then with a very high velocity actually push, what happened with a very high velocity of 1400 feet per second, what happened, 1400 feet per second, remember that. So with 1400 feet per second, this gene of interest with the gold or tungsten metal, this will be forcefully pushed, injected inside the host, because it is a gene gun method. Okay, like the gun shot is done. So this way the high velocity gold or tungsten are shot actually, so that our gene of interest along with which is earlier coated with our gene of interest can be inserted, right? So what is that? This is known as the ballistic method. Remember that this is going to be very much important, right? So remember that, what is that? DNA coated with microscopic plates of gold or tungsten of size 1 to 2 micron are shot at a velocity of 1400 feet per second. Then it is possible that our gene of interest can be injected inside the host body. Next one, see, we have electroporation. Electroporation means electricity will be given. So first of all, that host that will be taken, that will be subjected to high velocity, a very high voltage. Okay, so whenever high voltage is given, then what will happen? Suddenly, for a short duration, whenever high voltage is given, what will happen? The cell, the host body will suddenly develop some pores, and whenever pores are developed, the gene of interest will be inserted. But as it is a time period, the time period is very for a short period of time, so that's why whenever it is creating the pore, suddenly it will also shrink. So our gene of interest within that time period will be injected inside the host body. So this is the electroporation method. So what is given? Host with the rDNA is injected into the host body with high voltage. Okay, so that is the electroporation process. And the last one, that is known as the microinjection. So in this microinjection, what will happen? As we know already what is that injection means, what is gun means. So gun, at an interval we will shot the gene of interest inside the host, and with that microinjection process we'll take a microinjection. Okay, directly like the injection does, same thing is happening, that with the help of an injection, very small injection, we will put our gene of interest inside the host body, right? So what is that? Micro needle of 1 to 2 micron is used to inject the foreign DNA directly inside the host body. So yes, that is the micro injection process. So what we have, these are the methods by which we can make a host a recombinant host, so that we can able to inject our gene of interest inside the host body. How many methods we have? We have the chemical method, gene gun method, electroporation method, and the microinjection method. Remember that what type of host are used? Normally the bacteria, then yeast, bacterial cell can be used, the yeast can be used, then animal cell or plant cell can also be used. But it will depend on the type of experiment we are going to do. Okay. So next one, eukaryotic host will be preferred for the eukaryotic protein. If we want the protein to be eukaryotic, then also we have to choose the host which is also need to be a eukaryotic organism. Now next one, normally if we want to inject the gene fragment DNA, it cannot be inserted inside the host body. Why? Because the DNA gene of interest, that is hydrophilic, it is acidic in nature, and the lipid membrane will not allow the nucleic acid to enter inside it. Okay, so for that reason we are going to have different methods by which we can able to inject our gene of interest inside the host body, so that the host will not die. Okay, if during that procedures, during this method, suppose the host dies after that, then it will be of no use, right? So that's why the process by which we will able to inject or we will able to insert the gene of interest, but the host have to be survived and it will do carry its own processes. So for that we have different methods. Otherwise, you can see heat and thaw method is there, temperature fluctuation, which is also known as heat and thaw method. Okay, so in that heat and thaw method, what we can do, we can increase the temperature so that the cell can expand, insert the gene fragment. Once again decrease the temperature, cell will shrink, and our gene of interest will insert inside the host body. Similarly, we have another way, that is by the addition of some divalentine like calcium ion, we can able to develop some small pores in the host cell wall, and we can able to inject our gene fragment. So different methods are available here. First of all, chemical method. So first, this in chemical method, what we can do, that this host cell, which is not preferably should have the cell wall, polyethylene glycol PEG is added. So in this PEG medium, the cell will develop some pores, and we can able to inject our RDA. Next one, gene gun or ballistic method. In this gene gun or ballistic method, at our interval, we will just short the gene fragment with high velocity tungsten or the metal like gold at a velocity of 1400 feet per second. We'll directly short, we will short the gene fragment along with the tungsten or gold particle inside the host body, and this way easily the gene fragment can be injected inside the host body. Next one, we have the electroporation process. In this electroporation process, we'll just subject the host body, and along with that the rDNA is also here. Within a very small period of time, if we can create a high voltage, okay, electricity, then what happen, the cell will expand for a short period of time, for a short duration, then our gene of interest will be inserted, and this way we can able to inject our gene fragment inside the host body. So what is that? That is the electroporation process. Next one, that is microinjection. So in this microinjection, what happened, that the micro needles of 1 to 2 micron is actually used to inject the foreign gene directly inside the host body. So this way we can make the host competent. We will able to inject our gene of interest, our gene fragment with the vector or without the vector. Actually preferably with the vector will be injected inside the host body, and we can make the host competent. We can make the host preferable. We can make the host which have our gene of interest so that it can carry out our experiments. So that's all about the the third tool, that is the competent host. We have completed our tools of genetic engineering. First one, I have discussed the enzyme. Second one, that is the vector, and the third one, that is the competent host. So these are the three types of tools we have discussed. In the next video, we will see the separation process, how actually the gene fragment can be separated. We can carry out our experiment, so that we will see in the next video. But this way we will complete the whole three types of tools. So that's all about it. I hope you understand. Thank you.